Assalamu alaikum and a very good afternoon to everyone present. I'm Hussein and I'll be your moderator for today's session. Welcome to a webinar entitled Roaming in the Wild, Natural Sciences. This webinar is organized by the PROSPEN Committee of the Institute of Teacher Education, Penang Campus. Ladies and gentlemen, we have with us today Dr. Yong Kian Tai, the coordinator and senior lecturer of the Ecology and Biodiversity Program of the Institute of Biological Sciences, Faculty of Science, University Malaya. Dr. Yong is also the curator or the coordinator of the herbarium or the botanical garden at his faculty. In addition to that, Dr. Yong is also a council member of the International Association of Bryologists and an EXCO member of Malaysian Cave and Karst Conservancy, Malaysia. Dr. Yong's areas of expertise are bryophytes, which are small non-vascular plants like mosses, liverworts, and hornworts. His interest and specialization also extends to terrestrial bryophyte ecology and ferns. Okay, before I invite Dr. Yong to start this session, I'd like to mention here that questions from the participants can be posted in the chat box. Ladies and gentlemen, without any further delay, I would like to welcome and cordially invite Dr. Yong to take us on a vicarious roam into the wild with some of his expertise and interests in the realm of natural sciences. Dr. Yong, would you please?
Okay, thank you, Dr. Hussein. Thank you for the long introduction about myself, and very good afternoon to everyone. Okay. I'm Yong from University Malaya. So today in this today in this 1 hour, about 1 hour discussion, I will briefly share with you all about my research experience. Okay, so sorry. Sorry for this. I have to restart my PowerPoint. Sorry.
No problem, Dr. Yong. These sort of things happen to the best of us. Don't worry about it.
Yeah, very sorry. Very apologize. Can you see my slide? Oh, not yet. I think. Okay, let me share. Windows. All right. I think you can see my slide. Okay.
Yes.
Okay. Very sorry for all those. Okay. I'm Yong. I'm from University Malaya. So today I'm going to thank you. Thank you for the institution for giving me this opportunity to share with everyone my research experience. As I put in the title, today we are going to talk more about natural sciences. Natural sciences, which is my main research interest. Okay, I am basically a botanist, a plant person, or a person who study plants. I study, the diversity of plants, the ecology of plants, and also the biogeography distribution of different plants. And my main research interest is actually into this group, the plants we call bryophytes, or in Malay, we just call it lumut. Okay, lumut. So I look, I will study the differences of these different lumut, try to identify them, try to recognize them. There are actually many different types of lumut or bryophytes we have in the country. Okay, for mosses itself, okay, among the bryophytes, we have three main groups. We call them mosses, liverworts, and hornworts. Mosses, liverworts, and hornworts. For mosses itself, there are around 500 over species, actually 520 species in peninsular Malaysia. Okay, so for the past 20 over years, my main research is into this plant group. But recently, I also expanded my interest to look into the ferns and flowering plants. Okay, my I think my how did it work? So my research, of course, partly involves lab work. Okay, so just like many other researchers, we spend time in the laboratory. Examine our samples. We may use some equipments to study our sample. But at the same time, actually, I spend quite a bit of time in the field or in the forest to obtain samples, to obtain different samples. So I yeah, I quite often participate in such kind of research expedition. We visited different forests, go to different area in the country. Of course, I don't stay in five-star hotels. Okay, we only have these very simple, basic facilities. Okay, but I visit the five-star forest. Okay, I have been to the many different forests, enjoying the green. Okay, and the water, okay, as well as the forest, of course. I also visit mangrove, okay, uh, cave. Okay, actually, no plants inside the cave. But if I'm going to a limestone area, there are some interesting cave. I will just take the opportunity to explore the cave and go and check some interesting cave creature. As you can see here, this is a cave cricket, which you will only encounter them inside the cave. Okay, but to study bryophytes, actually, I spend a lot of time in the highlands. In the highlands, I go to the different mountains. Okay, different mountains to visit the different islands, especially the mossy area. So, when you visiting this forest, beside of exploring the bryophytes, I also very often encounter the chance to meet. To encounter some. I have the chance to encounter some beautiful plants. For instance, on your right hand side, I think everyone know what is this. This is a beautiful orchid, Epigenium kalianum. Actually, this orchid species, endemic in Mount Uru Kali, okay? This mountain, Mount Uru Kali. And also on the left-hand side, you have Phalagatis stonei. This is a plant that related to Sendudo. I think most of you know what is Sendudo, a common reedy plant. But this Phalagatis stonei is a species endemic. Endemic means you won't find it elsewhere, but only that particular location in the world. Okay, so this Phalagatis stonei is a species endemic to actually to Genting Highland area. Okay, so yeah, through this kind of survey visit, I actually have the chance to bump into all these beautiful and interesting, especially endemic species that are only present in Malaysia but nowhere else in the world. Of course, I also visited the highest mountain in the country, the Mount Kinabalu. Okay, so up on this mountain, you will came across this type of vegetation, okay, which is the shrub and the scrubs. This type of vegetation, you won't find it in the lowland tropics. You can only see it in the summit of a very tall mountain, such as Kinabalu. And this type of vegetation is actually more common to the temperate country. Okay? So if you want to experience the kind of the feeling of visiting temperate, but because of pandemic, we can't travel, no worry, go to Kinabalu. Okay? But okay, of course, on such a high, tall mountain, of course, the diversity is very interesting. You wouldn't find, the diversity that you came across up there are very far different from those found in the lowland tropics. Okay, so of course, okay, to reach the destination, for instance, up on this hill, as you can see here, uh, if you would like to go up to this summit area or the ridge area to locate some interesting plant, in this example, this is Batu Cave. On the summit, you actually have this endemic palm, okay, endemic palm species, Maxburetia rupicola. Okay, Maxburetia rupicola, this endemic palm. Can you see my tiny cursor? Sorry, I should change the cursor into a bigger cursor. Okay, so this palm only found up on the summit. Of course, to reach the summit, it takes many hours of walking and hiking. Okay. So and very often in the field, I do a lot of walking, or we just have to do a lot of walking to reach the destination. And sometimes you just have to climb up a steep slope. Okay, you climb up a steep slope to reach the higher elevation. Of course, if you are lucky, there are some ready-made structure. You can walk up the steps, the staircase like this to reach the higher ground. Or if you are real lucky and you have funding, maybe you can get a helicopter ride. Okay. And so far, my first and only opportunity to take to take a helicopter ride is during the scientific expedition to Maliau Basin, Sabah. Okay, so we took this helicopter and it was sent us to the higher elevation of the area, then drop us there. We camp inside the forest for 2 weeks. For 2 weeks to explore the forest and collect many interesting plant specimens. Okay? So I mean, besides I myself go to survey, I also bring students out for survey. So we also, go out together with the students. We climb many different mountains, okay? And we also go down the deep slope or the lip gully to look for interesting plants. And sometimes the experience can be very challenging, very adventurous. Okay, you have to squeeze through two big rocks to reach the other side of the hill. And of course, after all this, efforts, you will have chance to have photo with many interesting plants, including the big tree, like you can see here, or with the endemic species. This is the example of Maxpuratia lupicola. Okay, you can have photo with endemic plant and then share it with your friend. Okay, which I believe many of your friends don't have this experience. Okay, so this is what we do. Okay, what we do and what we, as a field worker, what we enjoy. Okay, so, of course, you can also have photo with tiny creatures, okay, tiny invertebrates from the, I mean, then also have photo with beautiful scenery, okay, beautiful scenery. And of course, being a student, they always know how to enjoy themselves. This is during a field sampling trip to a river. Okay, you see the students, they're happy taking photos, here another group of students enjoying themselves in the water and taking photos. Okay. Uh, of course, besides fun, we spend most time seriously looking for plants. Okay, this is what we do. We will search for plants, harvest them, put them into a collection folder, then bring back to the laboratory for study. Okay, so we will search different location or different, we call it micro habitats, where the plants can grow. And for my case, because I'm looking for bryophytes, they're often tiny. They grow between the branches. Okay, some epiphytic on the tree trunk, and also there are some found at the fold of the big rocks, like you can see here. Okay, and when you search carefully, very often you will be rewarded to encounter some very interesting plant. In this example, at this fold itself, okay, I encounter or I collected the third collection for Peninsular Malaysia. This species, these tiny moss actually grow, survive in this fold. Okay? This is the Trichostomum brachydontium. Okay? This species has been reported in Langkawi, in Perlis, okay, in Langkawi, then the third collection from Gunung Kantan Ipoh. Okay, Gunung Kantan Ipoh. Okay, so through this many collecting, reporting activity, I also sometimes reporting new information for the country, or new interesting plants. For instance, this example, Cololegunia pseudostipulata. This is a very tiny liverwort, as you can see here. The size is just a few millimeter long, a few millimeter long. Of course, uh, to have a detailed look of the plants, you have to you have to prepare a slide and place it under microscope. Then you see the leaves, the cells, and you can see the leaves is just about 0.2 mm, okay, 0.2 millimeter, okay, very tiny plants. But this tiny plant itself is unknown to Peninsular Malaysia until year 2020, where me and my student encountered it in Batu Cave. Okay, in Batu Cave, we reported this as a new record for Peninsular Malaysia. Okay, beside of this Cololegunia, we also collected this is second Cololegunia species. Okay, beside of Pseudostipulata, we actually reported another new record for Peninsular Malaysia during that trip in year 2020. This is Cololegunia raduli loba. Okay, this plant, Cololegunia raduli loba. Again, this is a very tiny liverwort, as you may notice here, this tiny one, the one in pale color. Okay, this tiny liverworts is actually epiphytic on another bigger size liverworts. By the way, liverworts is one of the bryophytes. It's a tiny green plants that cannot produce flower. Okay, and liverworts, just like other bryophytes, they are actually very primitive. They actually are among the earliest plant groups that evolved originally in the water, but first come and colonizing our land area. Okay, colonizing our land area. Therefore, they are very primitive in terms of their structure. They don't have proper water and nutrient transporting system. The system you call vascular. Okay, this group of plant, they are lacking this structure. So in a way, they cannot channel nutrient and water efficiently inside their body, but they rely on the water and nutrients that present surrounding them. So they usually have a very thin structure, okay, very thin structure. Their body can absorb the nutrients and water directly through contact. Okay, through contact. If you have water, then you will be entering the cells, or through a process you call osmosis. You'll be diffused into the cells. So this is the type of plant that I study, and this is a new record for Peninsular Malaysia reported in year 2002. And in this example, you happen to see a female organ. This is a female organ of the plants, Cololegunia raduli loba. Of course, beside of new record, we also encounter very rare plants. Okay, in this example, this is a moss. Okay, this is a moss, Podium biseriatum. This is the scientific name. Sorry, I keep on mentioning the name, forgot to explain to you. This is the scientific name, okay, of the species. This is Podium biseriatum. If this is the plant itself, okay, of course, this is under the microscope. You can see the branch, the leaves, and their rhizoids. They actually have no roots. As I told you, they are primitive. They don't know how to develop roots, but they have these fine hair-like structures developed from the plants. We call it rhizoids for them to grab on the substrate. Okay, okay. Then this plant, Podium itself, is very tiny. If you compare with a 5-cent coin, I think nowadays we seldom use 5 cent. But if you still, you have experience with 5 cent, you can imagine how tiny this plant is. Okay, this is the plant, and this is the same plant, but after we wet it in water. Up here, you can see the leaves are more expanded. Okay, that's the good part to study bryophytes, because bryophytes, their structure are simple. You remember I told you that they absorb nutrients and water directly through contact, through contact or diffusion. So if you apply few drops of water, then the plants will absorb the water and expand it. Okay, expand it. But when you want to store the plants, you just dry them. Then they will shrink. Okay, they will shrink. So after they shrink, completely dry, then you can store them for long time. But whenever you want to study them, apply a few drops of water, then magic happens. The plants will expand and recover to its original look. Okay. But if you ask me whether the plant can still survive, I will tell you I'm not sure, because some of the desert bryophytes, they do survive. Okay, they do survive without water for many years. Actually, those that living in the desert. But for the tropical species, most of them rely on constant presence of water. But of course, they may be seasonal, lacking water for a few days or few weeks. But they will not kill the plants. After they receive the water, they will again expand to its original shape and doing its own, reactivate its metabolism. Right? So back to the story of this Podium biseriatum. Why I'm so fascinated by this plant? It was because this plant, it was first reported for Peninsular Malaysia, for Peninsular Malaysia in year 1981. Okay, in year 1981. But indeed, in that report, it didn't indicate any collections, okay, any collection of this plant. But even after that, no one collected this plant. We only know this species presence as a record, okay, in a scientific report. It's only until year 2020, me and my student, Mr. Chia, encounter a living sample, okay, encounter a living sample, collected a real actual sample where we confirm the presence of this species in Peninsular Malaysia. And after the trip, we did some, conservation assessment research. We did some conservation assessment on this plant species, and we give this plant a status of critically endangered in Peninsular Malaysia. Okay, so far we only know this plant from one particular location in the country. Okay, in the country, and only know there are only a few populations, that surviving population present there for the country. Okay, so we give this plant a status of critically endangered, right? So yeah, beside a podium, we also have other interesting record like Erythrodontium julaceum. Okay, this sample. We reported this as the second collection for Peninsular Malaysia. Okay, it was first collected in year 2007. Then we collected recently. Next one, Hymetostelium recurvirostrum. Okay, Hymetostelium recurvirostrum. Again, this sample or this plant representing the second collection for Peninsular Malaysia. Okay, this is the second collection for Peninsular Malaysia. And this plant was found to present on this rock. Can you see this big rock? Okay, it's only present here on this part of the rock below the statue of Lord Murugan. Okay, actually we are quite worried. Yeah, we're quite worried about this population. Okay, we did a search around the area. We did a walk around the area, try to look for other population, but we couldn't find any. Okay, we couldn't find any. Apparently, this is the only surviving population present here on this rock. And it happened that this rock is actually located at the entrance, okay, entrance of a popular tourist spot. It means there are a lot of people walking, passing by, visitor passing by, and there are a lot of human activities around there. Okay, our main concern is actually, will these human activities actually threaten this population? The second record of this plant for Peninsular Malaysia. Okay, of course, beside of a lot of assignment during the field works, we encounter many interesting, pretty plants. Okay, but also there are plenty of I say there are plenty of sad moments. For instance, which I'm going to tell you is this plant, Cyatodium carbonarum. Okay, which I actually collected this plant on this rock. Okay, on this rock, or you call it stalactite. Stalactite. So this rock, it was fully covered. Okay, fully covered this rock, especially the lower half. The lower half of the rock. This Cyatodium carbonarum. This plant was very abundant there. That was in year 2004. Okay, 2004. And when I went back to the place recently, I was shocked. I was stunned actually because I saw this. Someone erected, a small gerai. Okay, just a small gerai in front of the rock, it blocking the sun, blocking the water. And then I went to check the rock. The plant disappeared completely from the lower half of the rock. It may be some still present up here, but majority of them already disappeared. Okay, disappeared. That's what we field worker quite often encounter. We may have collected something from a particular location, but when we go back to that location again after a few years or after many years, and we see there was sudden, there may be some major change of the site, and then the plants disappear from that location. Okay? So just to give you more example, this rock wall, initially it was covered by many plants, something like this, very dense. There are many plants. But we human, because we go and occupy the area, we want it to look pretty and nice. We go and scrap all those plants off and then cover with cement. Okay, cover with cement. So we go and occupy the area which originally occupied by the plants or by the animals. But then when we arrive, we go and erase them. Yeah? We go and the human itself is very damaging. We go and remove all of them. And sometimes, who's know, involving some rare and threatened animal or plant species. Okay? So uh, yeah, just to give you more example, this is one of the one of my trip to one of the limestone hill in Ipoh. When you climb up the hill, when we climb up the hill, we get this nice scenery. Of course, this pond, the ponds you see here is actually an ex-mining pond, which they converted it for keeping some freshwater fishes. Then at this on this particular spot, you get some nice scenery, but you turn your head to the other side of the hill, you see this. Uh, I mean, I mean, the, this. I mean, people are extracting. People are blasting, extracting the cement. Okay, from the limestone hill. Okay, so we are losing our diversity. We are losing our nature. We are losing our diversity. And of course, this we know this is nothing new. Since if you look at this map, since 1954, we are constantly removing or trading our nature. Okay, our nature in a way we say for country development. That's fine. If you really use the money to help the poor to develop country, that's very fine. But I mean, it seems like there isn't a full stop for this activity. We constantly removing our rainforest. Even year 2012, as you can see here, we're still losing our forest annually, every year. And this is not just an issue for Malaysia, actually happened in the whole Southeast Asia, everywhere. Yeah, you can see the loss of the green covers from year to year. Okay. And yeah, even some interesting location like this one. This is Kantan Hill in Ipoh. Okay, on this limestone hill itself, we actually recorded many interesting rare and endemic species, both plants and animal. But the area was given to a private company for cement extraction. As you can see here, the hill initially was a whole area like this, but half of it has been blast. Okay, has been blast for cement. And the company actually they got the permission to blast almost the whole area except area D. Okay, actually they got the permission to use the area D as well, but the company in a way they say they will conserve area D. But here in area C, we have endemic and rare species. And here at the foot here of area C, we have the very rare, very, uncommon limestone forest. Okay, we have limestone here in the country, but we don't really have many limestone forests. Where in this location, we have this limestone forest. But to the company, They don't care about all this diversity. What they want is just the cement. Okay, the cement. Huh? So they were going to blast limestone forest as well as Area C, okay, for cement extraction. Okay, so there are always this type of conflict between the scientific interests, the people's need. We need green area, we need clean air, we need clean water, but also with the conflict of the corporate interests. Okay, so there's always this struggle of this different conflict of interest. And actually, not long ago, just end of last year, I believe all of you, even though you're not staying in Selangor, but you heard about the major flood that happened in Selangor. Okay, and just to let you know, this is actually an old photo. This was taken, I think, 7, 8 years ago during the flood happened that time. Okay, it means this flood in Shah Alam area or in Selangor is actually nothing new. It happened almost, I wouldn't say every year, but it happens quite frequent. We have this flood episode happen for a while already. But last year, that was very major, I would say, very major, affected many areas, and it caused a loss of human life. Okay, so yeah, if you look at the map, okay, why flood can happen, you will see all these rivers, okay, all these rivers, they will actually join, and the water from all this area will join, connect, and go into Klang River. Then the water will be channeled out into the ocean, okay, into the sea. Okay, but if you look at the upstream area, you can see they are made. I mean, this area, most of them has been developed. Okay, this is Kuala Lumpur, main Kuala Lumpur area. Surrounding it, all these tributaries. You look at the areas, most of them have been developed. Okay, have been developed. So, and up here, this is our main range. Okay, main range. Of course, the development is almost up to the foothill of the main range, and even on the main range itself, if you go to Bukit Tinggi, if any one of you have visited Genting Highland, Bukit Tinggi, you must have noticed that there are now a lot of vegetable farms on the slope. Okay, vegetable farm on the slope. On the other side of the main range, you have durian farms. So it means the development now is all the way until not just the foothill of the main range, but already up to the slope of the main range. Then you can imagine when it rains, what will happen? The water, of course, there's no more vegetation to trap this water. The water will enter quickly to the rivers, and it will be collected, okay, collected, then channel out through the main river. And whenever there's this bend of the river, or whenever the location is a bit low lying, definitely when you have huge amount of water from the upstream, the water will accumulate here. And this is Sri Muda, okay, where badly affected by the last rain, the flood episode. Okay, so yeah, we can say this was, how could this happen? This could be, I mean, this could related to the unplanned development. Okay, unplanned development. Of course, all this development is not current. It's not happened overnight. It has happened many years back. Okay, many years back, but no one stop it, or no one slow it down, or no one prevent it. Okay, uh, in a way, again, we human, we are actually causing a lot of loss in diversity. Okay, this tapir was knocked down by a vehicle. Okay, so human, I mean, uh, in a way, we human contribute significantly to the loss of the nature. Okay, all right. Okay, I think I still have time. So let me move on to the second session. I mean, second part of my talk. Okay, which is upon the request of Dr. Koo. So would like me to talk a little bit on the quantitative study or quantitative research. Yeah? So I've been sharing with you my experience in doing research related to the nature topic, where we do a lot of field works, we do a lot of collections, but eventually all these all this information collected in the WA, okay, specimen collected in the WA, we will study them, then we will sort it out, then come out with a report, okay, and all this report we will actually sum it up. We'll sum it up to come into the figure, then do some analysis, okay, do some analysis. That is the quantitative part, okay. These are a few of my students. They are doing sampling on the ground to study the plants or the grasses, basically are the grasses. So in order to obtain the data, of course, you have to conduct sampling. Okay, you have to conduct sampling. So you have to design your sampling in a way to give you meaningful data. Okay, so let us look at the sampling. There are various ways to conduct samplings, and the sampling method also rely on the organism or the object that you're going to study. Okay, just now these are students collecting plants. Okay, these are students collecting on your left. These are students who like to collect insects. So they set up this trap. If I'm not mistaken, this is what they call Malays trap. Okay, so this is to trap insect inside the net. And here on the right hand side, they set up some mist net. Okay, they are setting up some mist net to trap bird species. Okay, so different sampling, you require different sampling technique, okay, for different organism group, okay, organism group. But the more important is you must design your sampling, okay. So you must plan for an effective sampling. Okay, you can simply randomly sampling something, but eventually the data may not be meaningful. So you must plan your sampling carefully and must take into the consideration of three main factors, which is the time you have. Okay, it means the length of your sampling. Okay, how long you want to carry out the sampling, the cost you have, the money, the budget you have uh, to to operate your sampling, then the workforce you have, whether you yourself going to sample the information or you have a team, a group of people to help you to sample the information. Okay, and uh, yeah, I was told this sharing is we have both students from the science and the non-science background. But this part of discussion is actually general, apply to both science and non-science students. Even you are non-science student, either you are doing literature, you are doing economic, you will still conducting sampling sometimes. You need to sampling for information. Okay, you need to sampling for data. For instance, when you coming out with question A, that is the time you want to sample information. Okay, so you must still plan your sampling. You cannot simply just distribute your question A because the data you obtain may not represent the community, or the data you obtain may not represent the target group that you would like to study. Okay, so you have to plan your sampling. Then only then your data will give you some meaningful information. Okay, so just bear in mind three main factors that you need to consider: the time, the cost, and the the workforce. Sorry, the workforce in conducting an effective sampling. Okay, so what method to use or what approach to use? Of course, there are two main approach. One is random. The second one is non-random. Okay, random and non-random. So of course you can break down this sampling method, random and non-random, into many different 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 branches. I won't go into this in detail, which you may refer to your lecturer in future when you're conducting research. See which method is most appropriate to your case, or to your research. Okay, but let us just focus on the random sampling. Okay, random sampling, which is the most often. Okay, which is the most often. Why we call something as random sampling means we don't want, bias inside the sampling. Okay, we are the researcher, we conduct the research, but we don't want our personal interest to influence the data and the result. Therefore, we have to conduct it in a random manner. Okay, in a random manner. Okay, so yeah, just give you some example. This is what I do. Okay, what I do for my research. I go to different sites. As you can see here, I went to Batu Cave, Selangor. I went to Gunung Kantan, Perak, and these are report from other researcher, Gunung Senyum, Pahang, and Wang Kelian, Perlis. Okay, these are by other researcher. So we went there, we do sampling. After that, we process our specimen, we identify our samples, then we group our plants according to family, genus, and species. Then we come up with this summary. Okay, we go and count how many species we have, and they are belong to which genus, which family. So we come up with this number. Okay, we translate the information into quantitative data. Okay, we translate it into quantitative data, then we sum up. So we have this total number of diversity recorded at each of these location. Okay. Of course, no matter what method you use, during sampling, you will still get some data. Okay. But the key point, as I mentioned, is whether this data is meaningful or not. It relies on your sampling approach. Okay. With the data, with the quantitative data that you have, you can conduct some analysis. Okay, you can conduct some analysis. Here in this research, my objective is to understand the correlation or the similarity in terms of the diversity of these different locations. So I have Batu Caves compared with Gunung Kantan, Batu Caves compared with Gunung Senyum, Batu Caves compared with Wang Kelian. Okay, I would like to look at the similarity in terms of their plant diversity, or we call flora. Okay, the flora plant diversity. Yeah? So there are some indexes or some existing formula that you can use. In this example, I chosen Sorensen's similarity coefficient, or you can call Sorensen's index, and Jacket index. Jacket index. These two different indexes that established by other researcher. Then they have tried out in different experiment. So they find these two indexes are useful to indicate the similarity of the diversity of comparing locations. So this is what I do. So I have the result, which is percentage. This representing the similarity of these two locations, Batu Cave and Gunung Kantan. In terms of diversity, similarity is about 45.33%. Gunung Senyum, 59.17%. Okay. Again, just to emphasize, no matter what sampling method you use, you will get some data. When you have data, you can just run some analysis, then you will get some result. But whether your result is meaningful or not, whether this analysis is meaningful or not, again, it back to your sampling method. Okay, it back to your sampling method. An experienced researcher can easily pinpoint your errors. Okay, you can get some result. You can present your result just like your computer. You key in anything, it can generate some result for you. But as an experienced researcher look at the result, they will know whether you make any mistake. Okay, they make any mistake, and the all of this actually you can trace back to your sampling methods. Your sampling methods is not random. You have so much of your own bias inside there. Then your result will be very obvious. Okay, very obvious, showing that you have some. I mean, you actually manipulate. Okay, you actually manipulate the result. Which experienced researcher, by looking at your sampling method, they were able to identify. Okay, so of course, if the result has been manipulated, means the result doesn't reflect what actually present on site. Okay, just like you do question A, if you manipulate, you want the result to fit something your own, something that you would like to see, then it doesn't reflect what actually happened in the community. Okay, so therefore, a random and fair sampling method is very important. Okay, very important. So yeah, just come back to the example of Batu Cave and Gunung Kantan. This is Batu Cave. This is Gunung Kantan. Of course, from this image, they looks like quite similar in size. But I have to tell you, Batu Cave is actually 1 time larger than Gunung Kantan in terms of the land area. Batu Cave is 1 time bigger than Gunung Kantan. I mean the leftover Gunung Kantan, I would say, because the other side has been blasted. This part has been blasted. So if you look at the data I showed just now, the total diversity, I mean for moss diversity that I reported for Batu Cave is 51 species, but Gunung Kantan is just 25. So about the half. Okay, about the half. So is it the truth? Because Batu Cave is 1 time larger than Gunung Kantan. Therefore, it should be 1 time more diverse than Gunung Kantan. But actually, not not always the truth, and not always true. But over here, I use a random sampling method. So just randomly collect the plants from different side of this hill. So eventually, I get this result. Batu Cave diversity is about 1 time more than Gunung Kantan. But if you want to be more precise, more carefully, then you have to have equal, equal sampling intensity. We say equal sampling intensity. How do you do that? You may have to sampling in fixed land area, like what you can do here. Okay, so you can see this all this square. Okay, these squares, they are actually referring to the sampling plot. You just sample within all this square. Okay, within all this square, this is fixed size. The size that you use in Batu Cave should be similar to the size you use in Gunung Kantan. Let's say this plot is about 10 meter times 10 meter. This land area, this square is 10 meter by 10 meter. So the square you use in Gunung Kantan also must be 10 meter by 10 meter. And you have to sampling the similar set of diversity. So you may have to lay 3 plots. Each of these we call plot. 3 plot at the foothill area. Okay, 3 plot at the foothill area. 3 plot at the summit area. Okay, why 3? Because you need to replicate the sampling effort. So you choose 3. Yeah, and then 3 plots at the slope area. 1, 2, 3. So you're repeating the same in Gunung Kantan. Okay, you can do the same in Gunung Kantan. 3 plots as a foothill. Okay, 3 plots at the foothill, 3 plots on the summit area, and then 3 plots at the slope area. Okay. So if you do this, okay, now you have the similar sampling effort in Batu Cave and in Gunung Kantan. Then the result you can now compare. Okay, the result you can now compare. So then you get the result are more reliable. Okay, I would say in this way you get a more reliable result. So yeah, these are the few features you must take into consideration. Actually, again, it's 3 main factors or 3 main features. You have to be consistent in sampling this different location. Okay, so you use equal number, you look into the equal site conditions, okay, and then you measuring the similar parameters. For instance, if you are measuring the temperature, then you use Celsius, degree Celsius, then you just use degree Celsius. So if you are measuring the humidity, then you're measuring the humidity of these 2 locations. Okay, and the
Dr.
second picture, yes, sorry, Dr. Singh.
I have a couple of questions from the members of the audience.
Okay.
The first one is, can one research be biased or become influenced by other non-purposive sampling factors? Okay, that's the first question. The second one is, how far can different locations affect the results of your research?
Okay. Let me answer the second question first. You mean the location affect the result of the sampling?
Yes.
Of course, this is actually a question of what we call biogeography questions. Yes, location do play a role because at different location, you may experience slightly different climate or weather. Okay, so for instance, I think you are in Penang. Okay, in Penang, you are on an island. Am I right? Island are surrounded by water. So your weather in Penang Island maybe is more mild compared with the mainland Butterworth area, whereas maybe the more inland area, like is it Sungai Dua, the more inward of the Penang area, which they tend to be more dry compared with the Penang Island. So this location itself will actually give you different result. Okay, or you are comparing Kedah. Okay, Kedah. I think you're all closer to the north. You experience the slightly drier weather. Compare with Kuala Lumpur, okay, to the south. Your Kedah, and towards the south, you go to Kuala Lumpur, or you go to Seremban, or you go to Johor Bahru. Okay, in a way, Kedah, you're more influenced by your weather is a little bit monsoon in a way actually. But Kedah or Perlis is very obvious. You will experience the monsoon weather where you have about half a year dry season, about half a year wet season. But you compare with the south of Peninsular Malaysia, where we are more constant in terms of our climate. We are humid throughout the year. So different location give you different result. And the other reason of different location is because of the soil factors. Different location, you have different type of substrate. If you talk about limestone area, if you go to Perlis, there's a lot of limestone here. You then the soil itself are more limestone-like. You compare with the main range, you go up to the Banjaran Titi Wangsa, you get more granites. In fact, most of our peninsular lowland area, we are made of granites, granitic soils. So these are different substrate. It gives you different type of result. Okay, so this is about locations. So again, it's not so much about the distance. It's more about the unique character of the location itself. But of course, the distance also to do with the weather. Okay, if you are far from the equator or closer to the equator, they give you different type of weather. Back to, sorry, Dr. Hussein, can I go back to the first question?
The first question was, can a research become biased or influenced by other non-purposive sampling factors?
Yes, that could happen. That could happen. But if you are doing it randomly, okay, you're doing it randomly, so you should already uh, uh, if those factors are not obvious, okay, I mean those factors are not, detectable, okay, not they are very minor. If you do it randomly, actually you eliminate those factors. But if those factors are already obvious, means you already can identify, okay, they are very visible, those factors, then you can already avoid them. Okay, avoid them. You choose a different location or you choose a different community. Okay, so that is how you do it. Or in a way, in another way is you take into consideration of that factors, the very obvious factors, and you have a control. Okay, you have a control then you can compare. That's why when you do research, if possible, you need to have a control, and you need to do replicates. Like I explained to you just now in this research, I'm not just laying one plot in one kind of site condition. I will do three because I repeat. Okay, I repeat. I have the replicate. With the replicate, I can compare. Okay, I can compare. Then I will able to identify those other extra factors. Those are different approaches that we can use to uh, actually enhance our research.
Okay, Dr.
Do I
Yun,
answer
thank
the
you.
question? Yeah.
Yeah, yeah.
Yep. Uh, all right. So I come back to this part, which is actually towards the end of my talk already. So these are the three features. First, consistency. Second, you take into the account of the different diversity. Yeah, this actually answer the first question. You have to consider, you have to take into account the different diversity. So to include those you say non-influencing factors, because they are also representing part of the diversity. Then the third one, it must be comparable. Okay, it must be comparable under different site condition, under different community condition as well. So yeah, so you do sampling. My students do sampling. Okay, during the day using different approaches, during the night using different approaches as well. And sometimes you involve different instruments for targeting organism. Okay, so towards the end, yeah, I would like to emphasize this. You look at this photo, what can you see? You saw some foreign visitors. And for the foreign visitors, they actually appreciate our diversity more than our local. Yeah, sad to say this. So people will travel from other parts of the world, come to Malaysia to admire our nature. But we Malaysian, okay, do we care about our nature, or maybe we are all in the process to appreciating our nature? Okay, with that, I would like to end my talk. Thank you. And yeah, Dr. Hussein.
Okay, Dr. Yong, thank you for that interesting explanation. I was just wondering about a few things, as you were presenting your research and, showed us slides on all that you've done, which seems very interesting, by the way. Okay, I was just wondering, you, of all the things to study, right? Because as we know, uh, our tropical forests are so rich in biodiversity, right? What prompted you to choose and specialize in biophytes and ferns?
Okay, yeah. I have been asked for this question many times. Yes, that's true. Actually, it's actually a very simple explanation. It's just because people tend to ignore these tiny plants. So when we talk about nature conservation, protecting the nature, we have a lot of emphasis on the so-called icon species. Okay, of course, we put a lot of effort to look after our tiger, our rhinoceros, or maybe some big trees, beautiful plants, but we have very little attention, okay, almost no attention to things like bryophytes. Okay, bryophytes or ferns, which you just walk past, you just step on them. That's actually one of the main reasons why I choose bryophytes. Since no one care about them, then I care about them. Okay, it's just because of that simple fact. But in a way, I'm not saying that care about tiger is not important, especially now. We need a lot of emphasis to look into this tiger because the tiger population is running very low. I mean, perhaps many of you know that we only left around 200 or maybe less than 200 tigers in the country. Okay, and bear in mind that these 200 tigers, they are not living in the same forest. They are isolated and they are separated. They may not able to meet each other. They cannot reproduce. Then eventually we are going to lose all these tigers. We just lost our rhinoceros 2 years ago, year 2020. We lost our last rhinoceros in the country. Now rhinoceros is officially extinct from Malaysia. And that can happen to any other organism.
I remember reading about this in the newspaper, yeah, because the
Mm.
Malaysian government actually asked the Indonesian government to
Mm.
help, breed this,
Yes,
and they actually turned us down.
yes, it's true,
Yeah,
it's
it's
true.
sad,
But
isn't
but
it?
the issue is when we have too limited individual, the survival, the successful rate will be very low. You just rely on one or two individual. It's just like a tiger. You rely on these 200 individual. You want them to reproduce many. It's difficult unless you have thousand or more than thousands. Okay, so therefore, we need a lot of effort. We need a lot of effort to take care of the limited individual. We hope the limited one can continue to survive. And the best is if they can reproduce, then we have higher chance. Okay? So we can invest millions or billions of dollars or ringgit, but just to safeguard one or two individuals is very difficult.
I, Doctor, I was also wondering, yeah, when you were presenting your slides in the
Yes.
earlier part of your talk, right?
Yep.
Do pyrophytes and ferns serve any specific ecological function? And also, what would happen if our environment becomes depleted of pyrophytes and ferns? Like, would anything happen?
Oh, yes. Uh, to most of the people, we tend to ignore them because they are tiny. They are tiny, and they have less, function or value, direct function or value to human. But bryophytes, just as any plants, they carry out photosynthesis. They release oxygen. We are enjoying oxygen, FOC. We are not paying a single cent for oxygen. But which organism can give you oxygen? Okay, not your girlfriend, not my wife. Okay, so it's the nature, it's the plants. But these are the free service that are provided by the plants, just like the bryophytes. They supply oxygen, and as tiny plants, they they absorb a lot of water. They also help to retain water. Okay, in the forest, the bryophytes function just like a sponge. Whenever rain fall, they collect the water, or the morning dew, it will be absorbed by the bryophytes. They store inside their body and slowly releases into the ground. Okay, then this go into underground, became our fresh water. And by doing this, they're actually regulating the local climates. Regulating the local climates. They ensure the forest environment is not extremely hot, it's not extremely dry. Okay, so all these are the function that provided by bryophyte. As a plant as well, they trap carbon dioxide. They trap carbon dioxide, then they help in mitigate global warming. So these are more like ecological functions of bryophytes, but not directly to human, and therefore uh, we human tend to ignore them. Same to the ferns. They actually play quite similar role, but fern is slightly larger compared with bryophytes in nature. Yeah.
Uh, Dr. Young, based on the slide you showed about this guy setting up a stall in front of the stalagmite where all these liverworts, right? They all died.
Yes. Yeah.
Uh, okay. Now, since this is happening around our country, right? Is there any attempt or effort put towards creating awareness among the people regarding
Yeah.
this?
Thank you, Dr. Hussein. Yes, they are ongoing effort. Now we see actually there are more and more NGOs or organization, local community that talk about caring about our nature. Okay, actually, there's a very good, uh, good move. And we see this advancement in the last 10 years. We see this improvement. We get more and more people. And even the school itself, we see there are more green clubs. There are Club Pencinta Alam, all those emerging. And I must say, thanks to the teacher, and teacher actually play a very important role here to educate your kids, educate the school children, uh, teach them to appreciate our nature, because we are actually benefiting from the nature, but we seldom relate all those what we are enjoying to the nature. So teacher play a very important role here to link the nature and human, link the nature and human. And when we learn more, then we care more. eh? Tak kena maka tak cinta lah. That's the popular Malay proverb. So teacher play a role in introducing nature to the kids, and then we in future we get a nation who care about our nature. And Malaysia is one of the mega diverse country. Okay, that many of the other countries, people in other countries, they don't enjoy that much of diversity, unlike us. As I say, the foreign visitor, they will pay so much to come to Malaysia just to look at our orangutan, just to enjoy our Taman Negara. We ourselves in the country, Taman Negara is not far from us. We just can take a car ride to get to Taman Negara. We have the nature with us. So it's up to us to appreciate it. Yep.
Okay, Dr. Young, thank you for the explanation.
Thank you.
Yeah, looks like our session is drawing to a close. And before, is there any parting message that you'd like to give to our audience here today? Any message from you?
Um, Again, I believe I'm talking mostly to the audience. Most of them will be teacher one day. Again, uh, I thank you everyone for listening to my talk. But these all future teachers. You are actually the future of the country. Basically, you are going to educate, teach, guide many more young kids. You are the one who can sow the seeds. You let the kids appreciate our nature, uh, and we just need more and more people, because we're actually running short of time. We are experiencing this global warming. Okay, we have this flood episode. We have this erosion at the seaside, especially in Penang Island. Okay, we are losing our mangrove area. So we need more and more people to participate, to love and care about our nature. So I hope the audience, the teachers in future, you can work hard in this. Uh, I mean, can help, yeah, can help to educate a nation where really care about the diversity we have in the country. Yeah.
Thank you, Dr.
Thank you,
Yong,
Dr.
for
Hussein.
the
Thank you. Mm.
interesting and informative sharing session. Now, for one, have always known what moss is, and I've looked at moss and then this moss. Yeah,
Mm.
but your talk today has opened up, other ways of thinking about moss.
Yeah.
So I think that's really good. Okay, everyone. Yeah. Looks like our session is about to come to a close. So, on behalf of the PROSPEN committee and the Institute of Teacher Education Penang campus, I would like to extend my warmest gratitude for being here with us at this webinar today. Yeah. I'd also like to thank Dr. Hasnul and Dr. Hasnul Hisham Hashim, yeah, and Dr. Kubi Lee for being behind the scenes and helping us out with the technical uh, support and coordination, yeah, so that this webinar could be materialized today. Now, before we part, I'd like to share a quote from Jane Goodall. I think most of you might know her. She's a primatologist and an anthropologist who's done extensive work with chimpanzees, if I'm not mistaken. All right. Now, she said this, and this is something for us to think about. She says, you cannot get through a single day without having an impact on the world around you. What you do makes a difference, and you have to decide what kind of difference you want to make. Right? I think that's something for all of us to think about, and that's also something that I feel as parents and teachers we need to impart onto our younger generation. If not, I guess that things would be so bad that it'd be very difficult to control or remedy the situation. So, with that, thank you all and have a good evening. Thank you.
Thank you. I also like to take this opportunity to thank Dr. Hussein, and all Dr. Koo and Dr. Nasro and everyone for listening to my talk. Thank you.
It's been a pleasure. Thank you.